If you've ever had, say, a really nasty flu or maybe even just a deep splinter that got all red and inflamed, you know your body can mount this incredible defense.
It really is amazing. Millions of cells all coordinating.
Yeah, macrophages, T cells, neutrophils, they're all moving, multiplying, communicating across, well, essentially miles of tissue inside you, right? How do they coordinate that? A single, unified response.
That's the crux of it, isn't it? It's a massive organizational puzzle. If some bacteria get into your foot, how does an immune cell way up in a lymph node know exactly what it needs to do?
And how fast to multiply, or crucially, when to stop.
Exactly.
So that's our mission today. We're doing a deep dive into the immune system's ultimate coordination systems, cytokines. These tiny things, these messenger molecules, they basically dictate the what, the where, and the when of the whole immune response.
And it's all based on these fundamental principles of pathology, inflammation, defense.
Yeah.
You really can't understand immunity without them.
No, absolutely not.
I mean, if you want to grasp complex immune diseases or how chronic inflammation works, or even, the latest immunology drugs that cost a fortune, you have to understand the cellular language. It's foundational.
Totally agree. The big picture here, and why we're focusing on these little proteins, is that pretty much all immune responses depend on them. Doesn't matter if it's a good response protecting you from a virus or, a bad one where your body attacks itself in autoimmunity.
Right. It all comes down to these signals. They control induction, regulation, everything.
They're literally the language of life and death for our cells, telling them what to do, where to go, when to divide, when to, well, die off.
It's that critical.
Okay, so let's unpack this. Let's start right at the beginning. What exactly is a cytokine?
Okay, fundamentally, cytokines are proteins. They're secreted, generally quite small, low molecular weight proteins. And their job is basically to mediate communication between cells.
Like little messages being passed around.
Exactly. They stimulate and regulate what various cells do. Mostly leukocytes, white blood cells, but also the endothelial cells lining your blood vessels, the epithelial cells forming barriers in your gut or lungs, even cells in your bone marrow. They talk to a lot of different cell types.
We often hear specific names, right? IL-6, TNF, IL-12. Can you clear up the naming? Why interleukin?
Sure. Yeah, many of the ones we know best, molecularly defined ones, are often called interleukins, or ILs for short. That name, interleukin, it came about because initially researchers understood them as mediating communication between leukocytes, interleukin.
Between white blood cells. Makes sense.
Right. Now, we know they do way more than that. They can signal to brain cells, bone marrow cells, structural cells, all sorts.
Right.
But that original name stuck, partly because their primary role is still regulating the immune system's main players.
Okay. And how they act, their mode of action, seems really key for understanding how targeted the immune response can be. Are they like hormones flooding the whole body? Or more like, I don't know, little localized whispers?
Overwhelmingly localized whispers.
Yeah.
That's a good way to put it. Precision is absolutely critical here. The source material really emphasizes that the main ways they work are autocrine and paracrine.
Okay, break those down. Autocrine.
Autocrine means the cytokine acts on the same cell that produced it. It makes a signal and listens to its own signal.
Like giving itself a pep talk or instructions.
Exactly. It's a crucial self-stimulatory loop. We'll definitely talk about that with T cells and IL-2 later. It's fundamental for amplifying a response.
Got it. And paracrine.
Paracrine means the cytokine acts on cells right next door. It diffuses just a tiny distance through the fluid between cells and hits receptors on neighboring cells.
So very local communication, like neighbors chatting over the fence.
Pretty much. It means the immune conversation is usually happening at high speed, right, where the action is, in that specific bit of infected tissue or that specific little zone within a lymph node.
Yeah.
Highly targeted.
That makes a lot of sense for keeping the defense focused and avoiding, well, collateral damage elsewhere. Does that mean they never act like hormones systemically?
Well, rarely is the key word for normal function. Acting at a distance, which we call endocrine action, isn't their usual mode. But, and this is a big but, this is where things can go badly wrong.
How so?
In really severe situations like overwhelming bacterial sepsis, you can get a massive uncontrolled release of certain cytokines, especially TNF and IL-1. So much is produced that it spills out into the bloodstream in huge quantities.
A cytokine storm.
That's essentially what it is.
Right.
And when concentrations get high enough in the blood, they do start acting like hormones, hitting distant organs, the liver, the brain, bone marrow, blood vessels everywhere. That's when you get systemic effects like high fever, shock, widespread blood vessel leakage.
Yeah.
It's the localized system being dangerously overwhelmed and going systemic.
So built for local chats, but can turn into a global shout during a crisis.
Precisely. The pathology often lies in that breakdown of localization.
Okay, let's follow that thread. Let's start with innate immunity, the first line of defense. When one of those sentinel cells, like a macrophage or a dendritic cell, first spots trouble, how fast does this cytokine stuff happen?
Almost instantly. That's the hallmark of innate immunity. These cytokines are produced really rapidly after the cell encounters microbes or, and this is important too, substances released from damaged or dying host cells, what we sometimes call DAMPs, danger-associated molecular patterns.
So not just invaders, but signs of internal damage too.
Right. It's like the body's first emergency broadcast, barrier breached, danger here.
And this first wave of cytokines, what's its main job?
Two core functions, really. First, induce inflammation. That means recruiting more immune cells to the site, making blood vessels leaky so they can get out, all that stuff. And second, especially if it's a virus, to inhibit virus replication. Put the brakes on the invader spreading.
Okay, who are the main players in this early cytokine response?
The major ones in innate immunity are TNF, that's tumor necrosis factor IL-1, IL-12, the type I interferons, or IFNs, another one called IFN gamma, which we'll see bridges innate and adaptive, and then a whole family called chemokines.
Okay, hold on, chemokines. You listed them as inflammatory cytokines. What makes them special?
Ah, right. Chemokines are technically a subfamily of cytokines, but they have a very specific job. Their name tells you chemo for chemical, kinesis for movement. They create chemical gradients.
Like a scent trail.
Exactly like a scent trail for immune cells. When inflammation kicks off, cells at the site release chemokines. These spread out, forming a gradient, and leukocytes like neutrophils and monocytes have receptors for them. They literally follow the chemokine trail, moving towards the highest concentration, which guides them right to the source of the infection or injury.
So they're the immune system's GPS navigation system.
That's a perfect analogy. They handle the traffic control, making sure the right cells get to the right place at the right time.
And who's actually producing these first signals? Who are the initial broadcasters?
The main sources, the heavy lifters, are those frontline sentinels, macrophages and dendritic cells, especially once they've gobbled up some microbes, but also specialized innate lymphocytes like ILCs, innate lymphoid cells, and NK, natural killer cells, are key producers.
Not just the classic immune cells, then.
No, and that's important.
Ah.
Structural cells, like the endothelial cells lining blood vessels or the epithelial cells forming your skin or gut lining, they can also pump out these cytokines when they're directly injured or infected. It's a distributed system.
Let's zoom in on IL-1 and TNF for a moment. You mentioned they can cause systemic effects like fever, and you hinted IL-1 has a really interesting activation story.
Yes, the inflammasome. It's fascinating. IL-1 is absolutely crucial for acute inflammation. It drives leukocyte recruitment, helps activate them, and yes, it's one of the main cytokines that travels to the brain to cause fever. But the key thing is IL-1 isn't just made and immediately secreted. It's made as an inactive precursor, pro-IL-1 beta.
Okay, so it needs to be activated. How?
Through this amazing molecular machine called the inflammasome. It's a multi-protein complex inside the cell cytoplasm.
What triggers it to assemble? What's it sensing?
It's designed to sense danger. This would be microbial products leaking into the cytoplasm, but importantly, it also senses signs of cellular stress or damage, things the body interprets as danger signals.
Like what kind of danger signals? Not necessarily microbes.
Exactly. Things like uric acid crystals, which can form when cells break down messily, or ATP spilling out of a damaged cell. ATP should be inside cells, not floating around outside. Even environmental irritants like silica or asbestos can trigger it. And one really interesting general danger signal is the loss of potassium ions from the cell.
Loss of potassium. How does that signal danger?
Well, healthy cells maintain a very high concentration of potassium inside compared to outside. If the cell membrane gets badly damaged by trauma or certain toxins or pathogens, potassium leaks out. The inflammasome sensor, often a protein called NLRP3, detects this drop in intracellular potassium and interprets it as a sign that the cell is severely compromised. It's a nonspecific but very urgent distress signal.
Wow. So it's not necessarily seeing the enemy, but seeing the internal chaos the enemy or just damage is causing. Potassium leakage as a generic help signal.
Precisely.
Yeah.
So when NLRP3 or another sensor detects one of these danger signals, microbial components, crystals, ATP, low potassium, The whole inflammasome complex rapidly assembles. It includes the sensor, an adapter protein, and crucially, an inactive enzyme called caspase-1.
And caspase-1 is the key.
It's the molecular scissors. The assembled inflammasome activates caspase-1. And activated caspase-1's main job is to find that inactive pro-IL-1 beta and cleave it. It cuts it, releasing the smaller biologically active form, IL-1 beta, which can then be secreted from the cell.
So no inflammasome activation, no caspase-1 cleavage, no active IL-1. A very tight control mechanism.
Extremely tight. Which makes sense, given how potent IL-1 is. You don't want it being released casually.
And this immediately makes you think about what happens if that control breaks down, if caspase-1 is firing all the time.
You've got it. That leads directly to diseases. There are rare genetic disorders called periodic fever syndromes or autoinflammatory syndromes, where people have mutations, often gain-of-function mutations, in the NLRP3 gene or related components. Their inflammasomes are basically stuck in the on position.
So they're constantly producing active IL-1.
Yes, even without any infection. This leads to recurrent, debilitating episodes of fever and widespread inflammation, essentially the body attacking itself because it constantly thinks it's in danger.
That sounds awful. But understanding the mechanism must have led to treatments.
Absolutely. It's one of the success stories of molecular medicine. Knowing that caspase-1 cleavage is essential led directly to the development of IL-1 antagonists, drugs that specifically block the activity of IL-1 beta.
And they work.
They work remarkably well for these autoinflammatory syndromes. Patients who were suffering constantly can have their symptoms dramatically controlled. It's a beautiful example of rational drug design based on understanding the pathway.
But this isn't just about rare diseases, is it? The source material connects the inflammasome to much more common problems.
Definitely. Think about gout, that incredibly painful joint inflammation. It's triggered when the inflammasome sensor proteins, the NLRs, recognize crystals of monosodium urate that have deposited in the joint. Classic inflammasome activation, massive IL-1 release, boom, gout attack.
Okay, so crystal recognition is key.
Yes. And it goes further. There's growing evidence that these same NLR sensors can recognize other types of crystals, like cholesterol crystals that deposit in artery walls during atherosclerosis.
Ah, linking inflammation to heart disease.
Exactly. Or potentially recognizing lipid metabolites associated with obesity and type 2 diabetes. The idea is that chronic low-grade activation of the inflammasome by these metabolic danger signals might be a key driver of the chronic inflammation underlying many common modern diseases. It's a really hot area of research.
Fascinating. Okay, let's switch gears with an innate immunity. What about viruses? They hide inside our cells. How do cytokines deal with that?
Right. Viruses pose a different challenge. You can't just eat them with a macrophage if they're hiding inside. This is where interferons, IFNs, are crucial, specifically the type I interferons like IFN alpha and IFN beta.
And they're produced when?
They're produced very rapidly by cells after they detect viral nucleic acids like viral RNA or DNA inside themselves. It's a direct response to viral infection.
And what do? Do they kill the virus?
Not directly kill the virus, but they induce what's called an antiviral state, not just in the infected cell, but importantly, in neighboring uninfected cells too. It's a warning signal.
An antiviral state. What does that actually mean molecularly? What changes in the cell?
It means the interferons trigger signaling pathways that activate a whole suite of genes. These genes produce enzymes that do things like degrade viral RNA or inhibit the machinery the virus needs to replicate its genome or make its proteins. Basically, the cell makes itself a very inhospitable place for viruses to replicate.
So it's like the cell locks down its own machinery so the virus can't hijack it.
Exactly. It's a preemptive defense. It might even trigger apoptosis, program cell death in the infected cell to prevent further spread. It's a really critical first line of defense against viral infections.
Okay. And the other big player in killing infected cells early on is the natural killer cell, the NK cell. How do cytokines fit in with them?
NK cells are amazing. They can recognize and kill virus-infected cells and even some tumor cells without needing prior sensitization, unlike T cells. They provide a crucial early defense, especially against viruses, before the adaptive response fully ramps up.
And they use cytokines, too.
Yes, they are a major early source of another key cytokine, IFN gamma. No, this is different from the type I interferons we just discussed.
Right. IFN gamma, we mentioned it briefly. What's its big role here?
IFN gamma is the single most potent activator of macrophages. Remember how macrophages engulf microbes?
Yeah.
Well, sometimes they need an extra kick to actually destroy what they've ingested, especially tough intracellular bacteria. NK cells, by releasing IFN gamma, provide that kick. IFN gamma signals the macrophage to ramp up production of things like reactive oxygen species and nitric oxide, powerful microbicidal molecules that kill the bugs inside the macrophage's phagosomes.
So the macrophage grabs the enemy, the NK cell shouts, "Kill it!"
Yeah.
with IFN gamma, and the macrophage gets the heavy weapons out.
That's a great way to think about it. Turns them into highly efficient killers. This is often called classical macrophage activation.
And is the NK cell activity itself regulated by cytokines?
Oh, absolutely. The whole system is interconnected. NK cell proliferation, making more NK cells, is stimulated by cytokines like IL-2 and especially IL-15.
Yeah.
And their killing function and their ability to secrete that crucial IFN gamma are strongly boosted by IL-12, which is often produced by macrophages and dendritic cells that have encountered microbes.
So IL-12 is like the go signal for NK cells.
Precisely. IL-12 tells the NK cells, okay, threat detected, activate fully, make IFN gamma start killing. It bridges the initial detection by macrophages DCs with the activation of the NK cell response.
This leads us perfectly into the adaptive immune system. Slower to get going, but much more specific, much more powerful in the long run.
Right, and here the main cytokine producers shift. It's principally the activated CD4 plus T lymphocytes, the helper T cells, that orchestrate things with cytokines.
And what are their main goals in using cytokines?
Broadly, three things. First, drive lymphocyte proliferation, make huge numbers of the specific T cells and B cells needed. Second, direct their differentiation, tell them what kind of effector cell to become. And third, activate other effector cells, like telling B cells to make antibodies or activating macrophages even more.
Let's start with that first point, proliferation. How does a T cell, once it recognizes its specific antigen, suddenly turn into an army?
This relies heavily on that autocrine loop we mentioned earlier involving IL-2.
Ah, the self-peptalk cytokine.
Exactly. So when a naive T cell gets properly activated, meaning it sees its specific antigen presented by, say, a dendritic cell, and it gets co-stimulatory signals.
The second signal, right, danger confirmation.
Right. Once it gets those signals, one of the very first things it does is start producing the cytokine IL-2, and it dramatically increases the expression of the high-affinity receptor for IL-2 on its own surface.
So it makes the message and the high-sensitivity receiver at the same time?
Precisely. The secreted IL-2 then binds to these high-affinity receptors on the same cell that made it. And IL-2 is a potent growth factor for T cells. Binding triggers intracellular signals that tell the T cell divide, divide, divide.
So that one initial T cell recognizing the threat rapidly becomes thousands, then millions of identical T cells specific for that same threat, clonal expansion.
Exactly. It's an exponential amplification process, absolutely essential for generating enough effector cells to actually fight off an infection spread throughout the body.
It makes sense that it's self-starting, this loop, but how does it stop? If IL-2 is such a powerful growth signal, what prevents T cells from just proliferating forever? That sounds like cancer.
That's a critical point. The system is built in breaks. Firstly, sustained IL-2 production and survival of these activated T cells requires continued antigen stimulation and those co-stimulatory signals from the antigen-presenting cells, APCs. As the infection gets cleared, the amount of antigen and activated APCs dwindles.
So the activating signals disappear?
Right. Without that continuous stimulation, IL-2 production plummets. Also, the T cells themselves become more susceptible to programmed cell death, apoptosis once the stimulation is withdrawn. And regulatory T cells, which we'll get to, also play a role in actively shutting things down. So the expansion phase is inherently transient, linked to the presence of the threat.
Okay, so T cells expand thanks to IL-2, but then you said cytokines direct their differentiation. They don't all stay the same, right? They specialize.
Correct. This is one of the most elegant parts of adaptive immunity. Depending on the type of threat detected, and crucially the types of cytokines present in the environment during their initial activation, those proliferating CD4 plus T cells will differentiate into distinct subsets.
Different flavors of helper T cells.
Exactly. Each subset is specialized, producing its own characteristic set of cytokines designed to deal most effectively with a particular class of pathogen. It's about tailoring the weapon to the enemy.
Let's break down the main subsets. What's the story with Th1 cells?
Th1 cells are the masters of cell-mediated immunity. They're primarily geared towards dealing with intracellular pathogens, microbes that live inside host cells, like viruses and certain bacteria. Think Mycobacterium tuberculosis.
And their signature cytokine is?
IFN gamma. We meet it again. Th1 cells are prodigious producers of IFN gamma.
And we know IFN gamma activates macrophages, so Th1 cells boost macrophage killing power.
Yes, powerfully. But it's often even stronger than the IFN gamma from NK cells. For maximal macrophage activation, what's called classical macrophage activation, you typically need both the IFN gamma signal from the Th1 cell and direct cell-to-cell contact. The Th1 cell expresses a molecule called CD40 ligand, which binds to CD40 on the macrophage.
Why the need for both? A sort of double handshake.
It adds another layer of specificity and control. It ensures that only those macrophages that are actually presenting antigen from the pathogen and are therefore in direct contact with a specific Th1 cell get the absolute maximum activation signal. When they get both signals, IFN gamma plus CD40 ligation, they become incredibly potent killers, churning out reactive oxygen species and enzymes to destroy whatever they've ingested.
Essential for clearing those tough intracellular bugs. But what if this powerful Th1 response is overactive or attacks the wrong target?
Then you get disease. Chronic Th1 responses are heavily implicated in many autoimmune diseases and other chronic inflammatory conditions where the immune system damages host tissues. For instance, the formation of granulomas, those organized collections of activated macrophages you see in diseases like tuberculosis or sarcoidosis. That's a classic Th1-driven process, an attempt to wall off a persistent irritant, but it often causes significant tissue damage itself.
Okay, so Th1 handles intracellular threats via IFN gamma and macrophage activation. What about Th2 cells? They handle different kinds of enemies.
Completely different. Th2 cells are specialized for dealing with extracellular parasites, particularly helminths, parasitic worms, things that are way too big to be eaten by a macrophage. They also play a major role in allergic responses.
And their cytokine toolkit is different.
Totally different signature cytokines: IL-4, IL-5, and IL-13.
Okay, what do these do? Let's start with IL-4.
IL-4 has several roles, but a key one is acting on B cells. It's the major cytokine that instructs B cells to switch the class of antibodies they're producing to IgE.
IgE, that's the allergy antibody, right?
Exactly. IgE antibodies are specialized to bind to receptors on mast cells and basophils. This arms these cells, readying them to release their inflammatory contents when they encounter the antigen again. This IgE mast cell axis is crucial for fighting worms and also for causing allergies. IL-4 also helps B cell growth and differentiation in general.
Okay, so IL-4 drives IgE. What about IL-5?
IL-5 is the eosinophil cytokine. Eosinophils are another type of granulocyte, particularly important for fighting parasitic worms. IL-5 promotes the production, maturation, activation, and survival of eosinophils and recruits them to the site of infection. Once there, eosinophils release toxic proteins from their granules onto the surface of the worm, helping to kill it.
So IL-4 gets the IgE ready, IL-5 brings in the eosinophils, and IL-13.
IL-13 has some overlapping functions with IL-4, but it's also particularly important for changes in tissues. For example, it stimulates mucus production by epithelial cells in the gut and airways, which helps physically expel parasites. It also contributes to airway hyperreactivity and asthma. And importantly, both IL-4 and IL-13 can induce what's called alternative macrophage activation.
Alternative, as opposed to the classical activation by IFN gamma.
Right. Instead of becoming potent killers, macrophages activated by IL-4, IL-13 become more involved in tissue repair, wound healing, and dampening inflammation. They can contribute to fibrosis or scarring in chronic settings. So a very different macrophage phenotype.
So the Th2 response, IgE, eosinophils, mucus, alternative macrophages, is beautifully designed to fight worms. But if it reacts to pollen...
Then you get allergic rhinitis, asthma, anaphylaxis. The same powerful mechanisms are deployed, but against a harmless environmental antigen, causing tissue damage and symptoms. It's the right response against the wrong target.
Makes sense. Okay, there's a third major subset that's been defined more recently. Th17 cells, what's their niche?
Th17 cells are critical for defense against extracellular bacteria and fungi, particularly at mucosal surfaces. They seem to handle bugs that aren't efficiently cleared by Th1 or Th2 responses.
And their signature cytokines.
Primarily IL-17, but also IL-22. Their development is driven by a different cytokine mix, too, involving things like TGF-beta, IL-6, and IL-23.
What does IL-17 actually do?
IL-17 is a potent recruiter, especially of neutrophils. It doesn't directly kill microbes much itself, but it acts on various other cells in the tissue, like fibroblasts, endothelial cells, epithelial cells, stimulating them to produce chemokines, those GPS signals again, and other inflammatory mediators.
So it amplifies the alarm call locally.
Massively amplifies it, specifically leading to a huge influx of neutrophils and monocytes to the site. These phagocytes are essential for gobbling up and destroying those extracellular bacteria and fungi. IL-22 complements this by helping to maintain the integrity of epithelial barriers and promoting repair.
So Th1 uses IFN gamma for intracellular bugs, Th2 uses IL-4/5/13 for parasites and allergy, and Th17 uses IL-17 for extracellular bacteria fungi via neutrophil recruitment. Highly specialized.
Exactly. It allows the immune system to tailor its attack strategy.
But you can't just keep attacking forever. All these responses cause inflammation and damage. How does the system put on the brakes?
Ah, yes, the resolution phase.
Yeah.
Absolutely critical. Uncontrolled immune responses would be disastrous. This is where regulatory cytokines come in, often produced by specialized regulatory T cells, TREGs, but also other cells.
The brake pedal.
Pretty much. They actively suppress immune responses and limit inflammation.
Who are the main players on the regulatory side?
The two big ones are TGF-beta, transforming growth factor beta, and IL-10.
How do they work? Let's start with IL-10.
IL-10 is primarily immunosuppressive. Its main targets are macrophages and dendritic cells. It inhibits their activation, reduces their ability to present antigens, and dampens their production of pro-inflammatory cytokines like IL-12. By shutting down these key initiating cells, IL-10 effectively turns down the volume of the whole T cell response. It helps terminate ongoing reactions.
So it silences the cells that start the whole process.
Yeah.
And TGF-beta?
TGF-beta is a bit more complex. It definitely has anti-inflammatory roles, particularly in inhibiting the proliferation and effector functions of T cells. It's crucial for preventing autoimmunity. However, TGF-beta is also a major player in tissue repair and remodeling.
How so?
It strongly promotes the production of collagen and other extracellular matrix components by fibroblasts. So while it helps shut down the immune attack, it's also involved in the healing process afterwards. But in chronic inflammation, persistent TGF-beta activity can lead to excessive matrix deposition, resulting in fibrosis or scarring, which can impair organ function. So it's a double-edged sword.
Okay, so we've seen cytokines initiating, directing, and terminating immune battles. But they have roles beyond immediate defense too, right? Like in just maintaining the system.
Absolutely. A key role is in regulating the production of blood cells hematopoiesis.
Right, you need a steady supply of immune cells.
Exactly. And this is where another group of cytokines, the colony-stimulating factors, CSFs, come in.
Like GM-CSF or GCSF?
Precisely. Granulocyte macrophage CSF, GM-CSF, granulocyte CSF, macrophage CSF, MCSF, and also cytokines like IL-3. Their names tell you what they do. They were discovered based on their ability to stimulate bone marrow progenitor cells to proliferate and differentiate into colonies of specific types of blood cells in lab cultures.
So they control the factory output in the bone marrow.
Yes. Their job is to boost the production of leukocytes, particularly neutrophils and monocytes macrophages, especially during infections when demand is high. If you're using up millions of neutrophils fighting bacteria, CSFs produced by cells like marrow stromal cells, T cells, and macrophages signal the bone marrow to ramp up production to replenish the supply. It's the logistics arm of the immune system.
Makes sense. Now, we touched on this with Th1 and Th2 responses, but let's circle back to the dark side. Tissue injury caused by the immune system in things like allergies or autoimmunity. How do cytokines drive this damage?
Well, the crucial point, as the source emphasizes, is that the mechanisms the immune system uses to cause damage in these hypersensitivity reactions are fundamentally the same mechanisms it uses to fight off pathogens.
The weapons are the same, they're just aimed wrong or firing uncontrollably.
Exactly. The problem isn't the weapon itself. It's that the cytokine-driven response is excessive, or it's poorly controlled, or it's tragically misdirected against something harmless like pollen, or even the body's own tissues and autoimmunity.
Let's use a specific example. Type hypersensitivity immediate allergy, like a bee sting reaction or peanut allergy. How is that a cytokine-driven disaster?
Okay, this is classic misdirected Th2 immunity. Remember, IL-4 driving IgE production?
Yes.
So during the first exposure sensitization, the person makes IgE antibodies against the allergen, say, peanut protein. This IgE then binds to mast cells, primarily in tissues. They sit there armed.
Waiting.
Now, the second time the person eats peanuts, the peanut protein allergens reach those IgE-armed mast cells. The allergen binds to and cross-links the IgE molecules on the mast cell surface. This cross-linking is the trigger.
What happens then?
Bang! The mast cell rapidly degranulates, releasing a whole cocktail of potent preformed mediators, the most famous being histamine, but also other things like proteases. It also quickly starts synthesizing other lipid mediators like leukotrienes and prostaglandins, and importantly, more cytokines like TNF.
And this chemical explosion causes the symptoms.
Instantly. Histamine and the other mediators cause blood vessels to dilate and become leaky, leading to swelling, edema, and redness. They cause smooth muscle to contract, which in the airways leads to bronchoconstriction, difficulty breathing, and in the gut can cause cramps and diarrhea.
If it's localized, maybe hives. If it's systemic,
If enough mast cells degranulate throughout the body, you get systemic anaphylaxis. Massive vasodilation leads to a catastrophic drop in blood pressure. Shock. Severe bronchoconstriction can close the airways. It can be fatal within minutes. All driven by that initial IgE cross-linking and the subsequent mediator and cytokine release, a defense mechanism gone terribly wrong.
it really highlights how critical precise cytokine regulation is. But this deep understanding, it hasn't just shown us the problems, it's also given us tools to fix them, right? Therapeutic breakthroughs.
Absolutely. This is where the story gets really exciting from a medical perspective. Understanding these cytokine pathways has revolutionized treatment for many inflammatory and autoimmune diseases.
So we can target these messengers now. Let's talk about inhibiting cytokines first. What's the poster child for this approach?
Without a doubt, the development of TNF antagonists. This is really considered a landmark achievement, a truly elegant example of rationally designed, molecularly targeted therapy.
For diseases like rheumatoid arthritis.
Exactly. In rheumatoid arthritis and other conditions like Crohn's disease or psoriasis, TNF is a major driver of the chronic inflammation and tissue destruction.
What exactly is TNF doing in, say, an arthritic joint that makes blocking it so effective?
TNF does multiple things. It promotes inflammation by recruiting more leukocytes and activating them. But critically in joints, it stimulates cells like synovial fibroblasts to produce enzymes called matrix metalloproteinases, which chew up cartilage. It also strongly promotes the formation and activity of osteoclasts.
The cells that break down bone.
Yes, so TNF is directly driving both the inflammation and the irreversible destruction of cartilage and bone that characterizes rheumatoid arthritis. By blocking TNF with antibody-based drugs or soluble receptors, you inhibit both processes. The clinical results for many patients have been dramatic, halting disease progression in a way older drugs couldn't.
And that success paved the way for targeting other cytokines.
It really did. Once TNF blockers proved the principle, researchers developed antagonists for other key players. We now have effective drugs targeting the IL-6 receptor, crucial in systemic inflammation and some forms of arthritis. There are drugs blocking IL-17, which have transformed the treatment of psoriasis and psoriatic arthritis. Blockers of IL-12 and IL-23 are used for Crohn's disease and psoriasis. IL-1 blockers, as we mentioned, for autoinflammatory syndromes. The list keeps growing. It's all about identifying the key cytokine driving the pathology in a specific disease and selectively neutralizing it.
So we can block them. Can we also use cytokines therapeutically, like give them to people to boost certain responses?
Yes, that's the other side of the coin. If a disease involves insufficient production or function of a particular cytokine pathway, we can sometimes administer that cytokine.
What are the main examples?
The most common use is probably administering those colony-stimulating factors like GCSF, granulocyte CSF, to patients undergoing chemotherapy. Chemo often wipes out the bone marrow's ability to produce white blood cells, especially neutrophils, leaving patients highly vulnerable to infection. Giving GCSF tells the remaining bone marrow stem cells to specifically ramp up neutrophil production, helping patients recover their counts faster.
So boosting the supply chain.
Exactly. Another example is using type I interferons like IFN-alpha to treat certain chronic viral infections like hepatitis B or C or some cancers. The idea is to boost that antiviral state we talked about, though administering interferons often comes with significant flu-like side effects because you're activating those pathways systemically.
Right, the systemic shout again. And you mentioned one more really important clinical use involving CSFs, mobilizing stem cells.
Yes, this has been transformative for bone marrow transplantation. Traditionally, getting hematopoietic stem cells required drilling into the donor's hip bone, quite an invasive procedure.
Ouch.
Yeah. But researchers discovered that if you treat a potential donor with high doses of GCSF for several days, it actually cocoses the hematopoietic stem cells to leave the bone marrow niche and spill out into the peripheral bloodstream.
The cytokines push them out.
Essentially, yes, they mobilize into the blood. Then you can collect these circulating stem cells from the donor's blood using a machine similar to dialysis called apheresis. It's much less invasive for the donor. These collected peripheral blood stem cells can then be infused into the recipient. Cytokines literally changed how stem cell transplantation is done.
Incredible. Okay, let's try and wrap this up. This has been a whirlwind tour of the immune system's communication network.
It really has.
We started by defining cytokines, these small secreted proteins acting mainly locally as autocrine or paracrine messengers.
Right. Then we saw their incredibly rapid role in innate immunity, things like IL-1 and TNF driving acute inflammation triggered by that fascinating inflammasome complex sensing danger. And type I interferons, setting up the antiviral state, with NK cells providing early IFN gamma for macrophage activation.
Then we move to the adaptive system, seeing how IL-2 drives that crucial T-cell proliferation via an autocrine loop, and how different cytokine environments push T-cells to become specialized subsets. T1 using IFN gamma for intracellular bugs, Th2 using IL-4, 5, 13 for parasites and allergies, Th17 using IL-17 for neutrophils against bacteria and fungi.
And crucially, the breaks regulatory cytokines like IL-10 and TGF-beta that are essential for shutting down the response and preventing autoimmunity.
We also touched on their housekeeping roles, like the CSFs managing blood cell production, and saw the devastating consequences when cytokine networks go awry in hypersensitivity, but also the immense hope offered by targeted cytokine therapies.
Yeah, blocking TNF, IL-1, IL-6, IL-17, it's changed medicine. And using CSFs or interferons therapeutically.
So the big takeaway for you listening to this is that cytokines are what turn the immune system from just a collection of individual cells into this incredibly complex, dynamic, coordinated network. They ensure the right cells are activated, the right weapons are produced, everything gets to the right place, and just as importantly, that the whole thing shuts down properly afterwards.
Which brings us to maybe a final thought to leave you with. We talked about the inflammasome, IL-1, and how understanding that one pathway linked things like rare fever syndromes to common diseases like gout and potentially even atherosclerosis and diabetes.
Right. Recognizing those danger signals like crystals or metabolic stress.
Exactly.
Yeah.
So the provocative question is, what other common chronic diseases, maybe things we don't even currently think of as primarily inflammatory, might actually be driven deep down by a subtle chronic dysregulation in a specific cytokine pathway that we just haven't identified or targeted yet? Think about neurodegenerative diseases, metabolic syndrome, maybe even aspects of aging. Could there be a key cytokine messenger orchestrating pathology there, just waiting to be discovered and potentially modulated?
A hidden language we still need to fully decipher.
Precisely. This tiny network of messengers likely holds secrets far beyond just fighting infections. It's fundamental to tissue homeostasis, repair, and potentially many chronic diseases of modern life. There's still so much to learn.